So up until now, all we've discussed really is what's DNA like, what's RNA like, what are they made of, and how do I get copies of DNA so that I could do cell division. But we need to move on to the next step. How does DNA actually make something happen in any of my cells? And the way that DNA does this is by directing protein synthesis. So just to make sure everybody is clear, an amino acid is the subunit or the monomer of proteins. So proteins are a big long chain, which is a polymer, so a big long chain of a bunch of repeating units, and the subunits are amino acids. The genetic code is the order of nucleotides in your DNA. And what we'll see is that the order of nucleotides in your DNA determines which amino acids form a protein. So when your cells are trying to make proteins, they're not just doing it randomly. They are taking instructions that come from your DNA. And the order of nucleotides in your DNA gets converted to the order of amino acids in a protein. So that is where that is coming from. Another term that we'll use to represent this is the word genetic expression. If you think about the word expression, it's what you see. So if I said, oh, your expression makes me think that you're sad, you've probably got, a pout or something on your face. It's something visible. And so we're trying to explain how is something visible, something that we can see, made from your DNA. So genetic expression is generally the process where your DNA gets converted to RNA, and then your RNA gets converted to proteins. Now there are two arrows, and each arrow represents a process. This first arrow represents a process called transcription. Transcription is when DNA is copied to RNA in the nucleus. So it's fairly important that you know where this is happening. Your DNA, as a general rule, stays in the nucleus of your cell. It doesn't go anywhere. Now, of course, if I was talking about a cell that doesn't have a nucleus, like a prokaryotic cell, of course, we wouldn't be able to say it was in the nucleus, but since generally we're focusing on what happens in a eukaryotic cell, this is where this takes place. The second arrow represents the second process, and that's the process of translation. During translation, we are going to take RNA and convert it into amino acids, and this is going to take place in ribosomes. Ribosomes can exist in more than one place inside of a cell. Ribosomes can be just sort of loose, not attached to anything, and so we would say that this happens in the cytoplasm. So it could be a ribosome just floating around. It could also be attached to the rough endoplasmic reticulum. So if you remember back to learning about plant and animal cells, probably back in Science 10, the rough endoplasmic reticulum is a bunch of folded-looking membranes that surround the nucleus. So if you take a look at the picture, it's representing this basic idea. In transcription, I am getting a molecule of RNA made, And in translation, I am getting what we will call, and I'll write it bigger so everyone can see, a polypeptide. A polypeptide is what we call a protein before it's finished. So a polypeptide is just a bunch of amino acids put together, and eventually it will get long enough and be called a protein. You'll see the word polypeptide used especially in exam questions because proteins are way too big. We could not expect you to look at an entire protein and talk about how it was made because it's too long. So generally in exam questions, we'll focus on just a little section of a protein to ask you about, and so you'll see the word polypeptide. Now, does anyone want to ask any questions about this overview? Because that's all this is so far, is the overview. Yes. How many amino acids? That's actually the thing that I'm going to say next. So if you wouldn't mind, I'll just wait till I go to the next slide. Does anyone else have any questions they want to ask here? So the next slide will answer the question that Shay just asked. How many amino acids go with your RNA? We need to talk about something called a codon. A codon is a sequence of 3 nucleotides of mRNA, and the little m stands for messenger. So we saw that there were 3 types of RNA. Specifically, mRNA is the type that gets made in your nucleus and that is going to come out into the cytoplasm. Each 3 nucleotides of your messenger RNA correspond to 1 amino acid. So it takes 3 DNA bases to make 3 RNA bases, and that will convert to 1 amino acid. Now there are 3 words that we would use to describe these codons that are making up your genetic code. The first word is that they are a little bit redundant. And so to explain this, I'm going to refer to this table. And I would like... So when I look at this table tells me which codon, so which 3 mRNA bases or nucleotides will code for each amino acid. And the first word I said we could use to describe it is that it is redundant. That means that the same amino acid has several codons. So if I were to look at this table, for example, you might notice right away these first 2. There are 2 different codons right at the top that would give me the amino acid named phenylalanine. I could have UUU, or I could have UUC. Either of those sequences will end up producing phenylalanine. Some of them have more. For example, down here, there are 4 different codons in that box, plus 2 up here, all that would give me leucine as my amino acid. So it's redundant, and we'll see in a couple of lessons how this is sometimes helpful. What that means is that if one DNA base were to change because of a mutation, you might not actually change the amino acid that's being produced, and thus there would be no effect in the person. The second thing about the genetic code is that it is continuous. So there are no spaces. Now that doesn't mean that every section of your DNA codes for something specific, because we saw that there were other types of genes. What we mean is that your genetic code is a continuous piece of mRNA. Some of them are coding for a protein, some of them are not. The third one, putting a little star next to it, The third one is that the code is universal. All living organisms have basically the same type of code. I would say all with a little star because there are a couple of exceptions and it depends on what you consider living and what you consider non-living. For example, viruses are not considered living because they require a host cell. But for all intents and purposes, all living organisms have the same code making up their genetic material. So a bacterial cell, for example, has the same nucleotides making up its DNA as you do. And the reason I'm putting a star here is because there is a lot of technology associated with this idea. One example that I could give you is that there are bacteria that produce insulin, which is fairly convenient. Insulin is something that we need to make for people who are, say, diabetic and require it. But because bacteria are made of DNA, and so are we could take the sequence of nucleotides that codes for the insulin protein, put it in a bacteria, and get the bacteria to make insulin. And this type of procedure is actually used fairly frequently in biotechnology to make lots of a substance that we require a lot of. If we were to try to extract insulin from a human, A, there are ethical concerns about experimenting on people, but B, it takes a long time. Bacteria reproduce really quickly, and so since we have the same code, we can make the same proteins if we have the same sequence of nucleotides. Now, when you take a look at this chart, most of the time charts are set up this way. This is the only version of the chart that you will ever have to use. It's the one that comes with your diploma. If you're looking at other sources, sometimes the way they put the titles is a little bit different. The first base is always listed along the left. The second one is always listed in the middle. And the third base is listed along the right. So if I had, let's say, ACU as my code, I would look down here and find A. That's my first base. So I know I'm in this row. Then I would find C as my second base. So I know I'm in this box. And then I would find U over here, so I know it's the top line. So ACU would be threonine. If you don't like looking at the sides, you can just look at the codes inside the box. They're always printed there as well. The other thing I want to mention is this little note that's at the bottom. And sometimes people ignore the notes because they think that they're not important, but in this case, they actually are. One of the codons, AUG, is called an initiator codon. Basically, this is the start codon. This is how your cells know, aha, the protein starts here. This is where I should start reading the code to make the protein. So it's called an initiator codon. I like to write the word start. The second note mentions 3 other codons that are called terminator codons. They're UAA, UAG, and UGA. They're all stop codons or terminator codons. They don't code for an amino acid. They tell your cell, aha, this is the end of this protein. Stop reading the code here and look for the next start codon to make the next protein. Does anyone want to ask any questions about that right away? So let's answer these questions together. First of all, can somebody tell me what does CCA code for? Very nice. How about AUG? Yes. Methionine. Anything else anyone wants to add for AUG? Anybody? Yes. Yep, that's what I got. Methionine. There's something else, though. So, methionine AUG is a start codon, and so I just wanted to make note of that. How about GCA? What does GCA code for? Yes. It should be glycine, unless I'm looking at this wrong. Yeah. Oh, I'm looking at it wrong. Sorry. I was looking at GGA. Now, question number 4 is those stop codons. So if you look either at the side of the table in your textbook or at the bottom of your table, you should know that they are UAA, UAG, and UGA. Now, the last question shows you the other way that we could ask you about this. A lot of the time we'll say which amino acid is being coded for. We'll go sort of in the forward direction. Sometimes, though, we'll go backwards and we'll say, this is the amino acid I have, how did I get it? So those would be the two ways that you would have to answer the question as well. So can somebody tell me, how would I get arginine? What would be one codon that would give me arginine? AGA would be one. How else could I get arginine? Yes. C, G, U. There are a few more. Anybody else got one? So if you take a look at your tables, there are 6 ways that you can get arginine. CGU, CGC, CGA, and CGG are all correct. And then in the box below it, there's AGA, which we already have, and AGG. So 6 different ways I can get arginine. So that means if I asked you what was the mRNA strand that got me this arginine, there are 6 possible right answers. So that would be a place where potentially more than one thing could be correct, depending on how the question is being asked. Does anybody want to ask any questions about this table? Okay. So we are ready to talk about transcription and translation now in a little bit more detail. Now, when we're taking a look at transcription, there are 2 strands of DNA, but we know that RNA comes in only 1 strand. So obviously, both of them are not going to be used. Only one of them is. Whoops, there's my red. So you'll notice that I have a couple of color-coded things in the picture, so that's how I'm going to refer to them. When I take a look at my DNA molecule, one of the strands is not going to be used as the template for RNA. It is just going to be there hanging out. The other strand is going to be used as the template for the RNA, which in this one is green. Now the sense strand is going to have the same nucleotide order as your mRNA. So if, for example, on this molecule of DNA, my nucleotide order was A, T, C, G, just to start. The other strand of DNA that I have would be complementary to that. So it would be T, A, G, C. Now, when I make my RNA, I'm only going to use one side of the DNA as the template. And anytime I'm making a new strand, I'm always making it complementary to the strand that it's matching up to. So when I make my mRNA, I would be putting A, U, C, G. So if I compare this and this, it's the same order. There is a difference, and the difference is U replaces T. So remember, in RNA, we have the same bases, except instead of thymine, we have uracil. So when I look at my mRNA, I have the same order as that strand on the top, the one that is not being copied. The other strand has the complementary base order to your mRNA. So it'll have the complementary letters. Now, it's not necessary that one side or the other of your DNA is the sense or the antisense strand. They can both be either. It just depends which side happens to get copied on a particular day at a particular time. So there's nothing specific about that top strand that means it has to be the sense strand, or about the bottom one that means it has to be the antisense strand. Now, when we're doing transcription, we are making a piece of mRNA. It's very similar to DNA replication. We have RNA polymerase. It does basically the same thing as DNA polymerase does during replication. Its job is to take new nucleotides and attach them so that I get a strand of mRNA. So it adds new nucleotides. Now, just like on the lagging strand during DNA replication, we need to have a promoter. So a promoter, just like it is in DNA replication, is a short segment of nucleotides indicating where to start. So it's a short segment of nucleotides, like a little sequence, that lets your cell know, hey, this is where we should start doing transcription. So we're doing transcription. RNA polymerase is your basic enzyme that is involved. You have two strands of DNA. One of them is just hanging out. The other one is being used to make a copy of RNA. The one that's just hanging out will have the same order of bases as your RNA. Does anyone have any questions that they want to ask here? I'll just remind you one more time. This happens in the nucleus of your cell. So anything that involves DNA should be taking place in the nucleus. Now, there are things necessary to induce transcription. It's not just going to happen just because any old time. When things happen out of control, that's how we get, for example, cancer happening, when your cells divide out of control. Transcription factors are generally environmental things. For example, hormones. So lots of hormones are produced in your body and the body of many organisms. We will talk about some hormones specifically as we get to the other units of this course. But several hormones that you produce will determine whether or not, say, your body is growing. So there is a hormone called HGH. It stands for human growth hormone. And that kind of hormone can determine whether or not transcription is taking place so that you're producing proteins so that you can grow. Now, environmental factors could be coming from your internal body environment, or they could be coming from the external environment. Water and temperature would be two other examples. If I were talking about seed germination, so whether or not a plant is going to start growing from a seed, the water availability and the temperature are both factors that could affect whether or not transcription is taking place. Now, there are tons of experiments that happen in regards to transcription. And so this might be a place where you might want to look for an article if this is something that interests you. The first presentation is coming up next week. There would be a lot of experiments, especially ones that involve not humans. It's hard to do experiments on humans, but on lots of other types of organisms, there is a ton of research being done on things that could affect transcription, environmental factors that would either start or stop transcription. Another thing that is often linked to this idea of transcription is apoptosis. This is programmed cell death. And so sometimes cells die on purpose so that other cells can multiply or other cells have more room to grow. And this could be something else that affects whether or not transcription is taking place. Now, another thing that I wouldn't consider environmental necessarily, because it has to do with the genetic code, but you have something called microRNA. These are short, usually they are 21 to 24 bases, which is really short if we consider that there are thousands of pairs usually in genes. But these little segments of microRNA can affect whether or not messenger RNA gets to be transcribed and then gets to continue on. So this microRNA can often repress mRNA transcription and or continuation into the next step. So there are short pieces of RNA that could prevent transcription from finishing or could prevent a transcribed piece of messenger RNA from moving on to the next step of the process. RNA with a little I, the I stands for interference, is simply the name of the process that this microRNA does. And so if you happen to see either of those abbreviations, they go together. RNAi is the interference that happens because of microRNA. So there are things that can start and things that can prevent or slow down transcription. Does anybody have any questions they want to ask here? There is also something called inverse or reverse transcription. There's a little picture here just so I had something to point to illustrate what was happening. Inverse transcription or reverse transcription is something that happens frequently with a virus. There will be an enzyme called inverse or reverse transcriptase. And this enzyme will cause the process to go in reverse, which is not typical. Normally, DNA goes to RNA, not the other way around. Inverse transcriptase is an enzyme that allows mRNA to be transcribed back into DNA and then be inserted into the host cell. So if I take a look at the picture, here's my virus. It's got its viral RNA. Through the use of the enzyme, reverse transcriptase, we can take that RNA and make DNA out of it. And then that DNA gets put into the DNA of the cell that the virus is infecting. And so this is a pretty common way that some viruses have their effect. Their RNA gets transferred back to DNA, and then their DNA gets inserted into the host cell as if it was part of that cell's DNA. So every time that cell reproduces, that viral DNA is getting reproduced along with the cell. You would not be expected to redraw this picture. I just wanted to have something to point to so that you could see what was happening. Does anyone want to ask any questions about reverse transcriptase? So we got one more transcription thing, and that's transcription modification. Frequently, once transcription has happened, or as transcription is happening, one of three things, or maybe all three things, will happen. The first thing is represented by this picture down here. It's called a poly-A tail. Poly means many. A is standing for adenine, the nucleotide. And so a poly-A tail is somewhere in the neighborhood of 200 nucleotides, all adenine, added to the end of mRNA. Now, why would your cell bother doing this? Frequently, enzymes that you have will actually break apart molecules in your body, maybe that you don't want it to. And so this is to protect from enzymes. So if an enzyme were to come along and start cutting the end of the molecule of mRNA, there's a whole bunch of extra adenines at the end that are not part of the code. So even if an enzyme were to attack the end of that molecule, the code itself is still intact. The second thing is a GTP cap. So the G here is standing for guanine, and the P is standing for phosphate. So we're adding some guanine and some phosphate to the end of RNA again. So it's added to the end of mRNA, and it's for the same reason, to protect from enzymes. So this way, both sides of your molecule of mRNA have some protection against an enzyme chopping a piece off and thus destroying the code. The third thing that can happen, which isn't pictured here because I've already shown this to you, is that sometimes introns will be removed. So excision means removing, and introns are those sections that do not code for a protein. So we're sort of cleaning up the RNA and only taking the parts that are actually going to make a protein. So your DNA is in your nucleus. It got copied into a piece of mRNA. That mRNA is going to travel to a ribosome so that translation can happen. Now, before we can talk about translation happening, we have to talk about something called an anticodon. It is a 3-nucleotide sequence attached to tRNA. T stands for transfer. So we saw a couple days ago there are 3 kinds of RNA. Messenger RNA is the one that's bringing a message. It's coming from your nucleus to the ribosome with the information the DNA had. tRNA stands for transfer RNA. If I were to loosely draw its structure, it's got this funny 3-lobed look to it. tRNA is still one single strand. It's just folded over on itself. On the bottom lobe, there will be the 3 nucleotide sequence. So just for fun, let's say GCG. Those 3 nucleotides that are at the bottom, so they're attached to the tRNA, they are complementary to your mRNA. So if that's my molecule of tRNA, the molecule of mRNA that came from the nucleus should have, in this little short section, CGC. So it's complementary. It's the opposite. It's the other part of the complementary pair based on what your mRNA is saying. Now the other thing that is attached over on this side somewhere is your amino acid. Now the amino acid that's attached is specific. It is based on this anticodon, which is complementary to your mRNA. So if you take a look at your little tables, ask yourself, if CGC is on my mRNA, which amino acid is that coding for? So if you look, if C is the first letter and G is the second letter and C is the third letter, we should be coding for arginine. So the transfer RNA will be attached to the amino acid that its anticodon complements. That's how the tRNA is set up. It'll always have the amino acid that matches the mRNA code that it will complement. Does anyone want to ask a question about that? Yes. That's correct. So it's the mRNA that came from your DNA. It's the one you read to know which amino acid you're getting. The tRNA always has the complement to the mRNA code. So mRNA tells you which amino acid. tRNA has the complement simply so it can come and attach in the right spot. Now the last type of RNA is ribosomal RNA. And I just want to just make a quick note about it. It is the RNA that is part of the ribosome. We're not going to look at its order because it's not going to determine the sequence of anything that's happening. So I'm just letting you know that's where it is. So in the picture on the right, you have this big structure in the background that has sort of 2 lobes. That's the ribosome. The mRNA that came from your nucleus is going to sort of go through the ribosome, kind of like how you would put paper money into a change machine. You put the paper money through the slot and it gives you coins. We'll imagine that the mRNA is the paper money, the ribosome is the slot, and the amino acids are coins. And that's basically what's happening. I'm putting something in, the machine is reading it, and giving me something else that's not the same, but that's equivalent to what I put in. So last thing I want to look at today is just a close-up view of what's happening during translation. Here's your tRNA. It's a much nicer picture than I drew, but it has the 3 lobes. Here are your anticodons. So this is an anticodon, that is an anticodon, and that is an anticodon. Here are your 3 codons, 1, 2, 3. So mRNA has codons that actually code for the amino acid. tRNA has anticodons that are the complementary base to what your mRNA has. And on the end of the tRNA, you have amino acids. And there will be an enzyme that will cause a chemical bond to form between the amino acids so that they will end up making one long protein chain. The cycle will continue until we get a stop codon.